We're going to have a presentation on the ornamental extension updates for Oahu and Hawaii counties by Russell Galanti. Russell is a junior extension agent for ornamental crops and landscape industries on Oahu and Hawaii Island. He is a graduate of the University of Hawaii at Manoa College of Tropical Agriculture and Human Resources, and he has worked in the green industry for 17 years. Russell works with the cut flower, nursery crop, turf grass, and landscaping industries through research and educational programs. Russell is currently working on projects related to pests and diseases in the green industry, new temperate cut flower trials, lowland protea variety releases, and new tree variety releases, as well as new marketing opportunities for nursery growers on Hawaii Island and Oahu. He is collaborating with other faculty at the University of Hawaii at Manoa, Hawaii Department of Agriculture, and multiple private businesses and organizations. Russell, welcome.
Good morning, Glenn. Thanks for the introduction. I appreciate the introduction. I'm going to go ahead and share my screen. So I'm going to be presenting my ornamental extension updates for Oahu and Hawaii counties. Just to give you an overview of what I'm doing right now and what I'm specifically going to talk about, I've got a list of my ongoing projects and new projects beginning hopefully in January 2021, funding prohibitive. So I'm going to be talking today on statewide survey for red ginger disease causing organisms. This is a project that I've been working on. This is something that I talked primarily about in the last HIFNA research webinar. So if you were there, you would have heard the updates from 1 year ago. I'm also going to talk a little bit about work that I had done on Podocarpus blight that was also discussed at the last research webinar or seminar. At that point, they were seminars, not webinars. I also have another project going, new germplasm. I'm working on dwarf potted sunflowers on Oahu. Not enough data to present on that and not enough time to present on that. Moving forward into January 2021, we've got protea variety trials and eucalyptus variety trials. And these are going to be going on Oahu, on Hawaii Island, and also statewide. So an overview and a recap on the red ginger disease complex. So this is a disease that started to be detected on Oahu Island with the epicenter in Kaneohe. At this point now, we've detected it statewide, and we have identified both viral and fungal pathogens as potential causal agents. We've also identified the fact that there are a mixture of viral combinations, often called a virome, in these gingers. And it's thought that these viruses are compromising the health of the plants that are then allowing one or more fungal pathogens to attack the plants and lead to decline, along with symptoms from the viruses themselves leading to declined symptoms. So there's all sorts of symptoms and characteristics for the multiple viruses that we have identified and are trying to characterize at this point, everywhere from streaking to mottling down to stunting and full-on plant decline, including loss of stems and necrotic tissue. We also see symptoms on flowers, including color breaking, as well as damaged finger-like markings on the flowers. By far, the biggest issue and the biggest symptom that we're trying to identify the causal pathogen of is stunting and dieback, because this also leads to loss in yield and overall crop decline. You can see from these pictures that there is a serious issue with these plants, which are in fact infected with viral and fungal pathogens. Usually plants are up to 8 feet, 10 feet tall when they're fully in flower, whereas virused plants, and we're going to go through the characterization of which viruses those are, tend to create a stunting symptom in these plants. And just an idea of what we're talking about close up in terms of fungal pathogens, most indicative of the fungal pathogen that we are experiencing in ginger is going to be leaf spot fungi, which I'll talk about a little bit. Unfortunately, I didn't get those slides into this presentation, so I'll just discuss them verbally at the end of the ginger talk. So far, viruses that we've identified, 6 viruses. First is banana bract mosaic virus. It's only been detected on Oahu samples. Then we detected bean common mosaic virus, a fairly uncommon virus in terms of our detection. Next, Canna yellow mottle virus. Now, this virus has been detected on all islands, and it's also one of the primary viruses that we've detected. So out of all the samples that we have collected and processed, Canna yellow mottle virus is one of the primary viruses that we identify. Another less important, because we don't see it as often, virus is banana streak virus. And then we've got 2 new viruses that have never been identified on ginger, and we don't even have them fully identified themselves. We do have them identified down to their viral families. Because we haven't published, we're not going to include the virus names on here. Now, virus A has been detected on all islands, and it is a virus that we have detected a lot. So it's something that we are seeing more and more of. Virus B has only been detected on Oahu, and we have not seen it on another island, although it is prevalent in samples on Oahu Island. So bean common mosaic virus, not one I'm going to talk about because we don't see it very often. Banana streak virus, also not going to talk about it very much because we don't see it that often. What we are going to talk primarily about is Canna yellow mottle virus, virus A, and a little bit of banana bract mosaic virus and virus B, just because they're not as common as the other 2, but their symptoms are severe in terms of how we've characterized them. So last year, I talked about identifying some of these viruses and some of the work that we've been doing, but we didn't quite characterize those viruses yet in terms of their symptoms. Now, this year, we've reached a point where we've had enough samples that we know what some of these viral symptoms are in terms of looking through all the samples, creating a spreadsheet of what has what symptoms, and trying to pinpoint the symptoms to each virus, which is difficult because there's often multiple symptoms. You're missing symptoms in certain viruses, from certain viruses in certain plants. It depends on the cultivar, the breed, the hybrid, or the species of plant. So I'm going to try to separate that a little bit in this talk. So first and foremost, we're mainly talking about red ginger, Alpinia purpurata. For Canna yellow mottle virus, we've primarily seen stunting and dark and light banding. So you can see on this leaf what I'm talking about with the dark and light banding. Yes? Oftentimes, in fact, every time we have Canna yellow mottle virus, we have seen symptoms of dark and light banding. So somewhat indicative of Canna yellow mottle virus, but as you see, it's also indicative of other viruses as well. So stunting is the biggest issue with Canna yellow mottle virus, and also overall. This is what you're going to see with a plant that is only infected in Canna yellow mottle virus in the field. You'll see the dark and light banding, and you'll potentially see stunting. Now, I have seen plants that are not stunted at all that have Canna yellow mottle virus. So it's potential that some of the symptomology of these plants is due to stress or environment. There is an exception. The cultivar Polynesian Princess has much more severe symptoms when only infected with Canna yellow mottle virus, including stunting, chlorotic mottle, marginal leaf yellowing, and then necrosis is where we start to see severe advanced symptoms, and then stem dieback. So these are a few leaves here that have just Canna yellow mottle virus, but you can see the difference, the chlorotic mottling here, the leaf yellowing that's going to lead to leaf necrosis and then stem necrosis. And here's a little bit of advanced symptoms in the field for that Polynesian Princess cultivar. So unfortunately, more susceptible cultivar. On the other end of the spectrum, there is an exception for the Darwin types as well. Fortunately, Darwin types don't seem to be as affected by Canna yellow mottle virus as the straight Alpinia purpurata. we only see chlorotic mottling, whereas they don't have stunting, they don't have decrease in yield, they don't have less vigorous plants. But we haven't seen a lot of canna yellow mottle virus in the Darwin types, so we can't say for certain that it will never cause stunting, just that in the samples that we've seen, we haven't seen stunting. So we're going to talk about virus A characterization, this mysterious virus that, don't worry, will become known once we reach a certain point in being able to fully characterize it. So you'll also see stunting and dark and light banding, just like canna yellow mottle virus. And they're pretty much identical in terms of symptoms in Alpinia purpurata. And in fact, here, take a look at this picture. We've got two sets of leaves, two different samples. Both have dark and light banding. So you can see the dark and light banding on the left-hand side, and on the right picture, you can see some slightly thinner dark and light banding. And this is on both immature and mature leaves. So leaves that are close to the ground and leaves that are close to the stem tips, including on flowering stems themselves. Now, this is a real problem. On the left-hand side, we were able to detect virus A. On the right-hand side, no positive results for any viruses, even though we are seeing the dark and light banding. Now, that can be due to the fact that we got a false negative, or that there's a virus that we haven't identified yet. So we're not picking it up in our sequencing. We don't have the genome for it. And this is what virus A looks like in the field most commonly. Now, fortunately for virus A infected plants, I have seen them be the most vigorous while still being infected with the virus. Now, of course, we've got viral combinations. I'm going to talk first about canna yellow mottle virus and virus A together in Alpinia purpurata. You can see a little bit more advanced symptoms in terms of the streaking, the chlorotic banding itself. And then in advanced cases, we're seeing marginal leaf yellowing and necrosis. Now, as a caveat, most of the plants that we detected canna yellow mottle virus and the virus A together were nursery crops. So they were plants that potentially were in not the perfect conditions. So these symptoms may have been caused due to stress or other environmentally related exacerbations. So virus B characterization. This one's a really interesting virus in some of the symptoms that it's causing in the different types of ginger. In Alpinia purpurata, your less advanced symptoms are going to be the stunting and banding, but you also see this really interesting leaf deformation, marginal leaf yellowing, marginal leaf necrosis, and of course, low production of stems and dieback. And this is kind of what you're seeing in the field in Alpinia purpurata. Some of the leaves tend to be deformed, they don't unfurl, and of course, stunting symptoms. Now, this is where it gets really interesting. There is an exception for the Darwin types where you see this very dramatic bleached chlorosis from virus B infected plants. And this is common for all the Darwin types that we have sampled for that have also been positive for virus B. And the symptom is bleached chlorosis marginally or throughout the whole leaf. And this is often the newest leaves are affected by this chlorosis, as well as leaf deformation. You can see how deformed these leaves are. Of course, low production of stems. You don't see any banding normally in these plants. You can see a little bit there, but that's not indicative of virus B infection. And now the plants tend to be less stunted and they still produce full-size flowers, but the low production of stems is a serious issue with this virus. And then of course, we can't stop there. We have to have an incredible mix of viral combinations. So we're talking virus B, virus A, and banana bract mosaic virus. And this is where it gets really bad for a ginger grower or for a ginger plant, because when we have combinations of 3 or more viruses, we see severe symptoms of plant decline. In the virus B and banana bract mosaic virus, the only symptom we didn't see was chlorotic banding, but we did see severe stunting and necrosis. And when you have all three, the only thing we haven't seen is bleach chlorosis. But to get to the point, if you've got 2 or more viruses and they are anything other than Canna yellow mottle virus and virus A, you're probably going to have a plant that is not going to recover from cultural practices to be a plant that you can actually produce flowers at an economic scale. So here's what we're looking at in terms of the combinations between virus B, virus A, and banana bract mosaic virus. A very attractive plant in some ways with the leaf variegation, but unfortunately the variegation and the flower breaking that was on this plant was caused by virus. Another picture of the same plant. So just to talk about the fungal pathogens for a little bit, I've got a mycologist that is working on the fungal work. What we did was we collected fungal samples from infected plants that looked like this image right here. So anything that had leaf spot and severe decline, we collected samples from. And what we did was we took isolates from those plants and plated those isolates. And the primary pathogens that we discovered were Nigrospora and Phoma. So 28 out of 58 of the isolates ended up being Nigrospora pathogens. 22 out of the 58 isolates ended up being Phoma pathogens. Now, Nigrospora is a typical leaf spot causing fungi. Phoma is also something that can cause spotting on leaves, but it's also a fungal pathogen that affects other parts of plants. Now, the problem here is that we haven't been able to re-inoculate either healthy or diseased gingers with these fungal isolates. We've got 4 fungal isolates for the Nigrospora that are different from each other, and we've also got different fungal isolates for the Phoma. Unfortunately, given 2 so far attempts to prove Koch's postulates, we have not been able to successfully do so with any of those fungal pathogens, even though they seem to be the primary candidates for the fungal disease causing agents portion of this mystery. So we're still working on that and moving forward with more pathogenicity testing. So in conclusion, we've sampled, this is a little bit older data, we've sampled a little over 66 samples. Banana bract mosaic virus was the most prevalent before HTS, high throughput sequencing, which is our method for identifying the viruses. Oh shoot, I didn't forget to take that out. Don't look at that. Virus A is the most prevalent virus after high throughput sequencing. 18 samples tested negative. Canna yellow mottle virus was detected from mealybugs. So we were also trying to determine what was the vector for Canna yellow mottle virus. And without being able to necessarily publish it, we have detected Canna yellow mottle viruses in mealybugs. What we're missing is revectoring by the mealybugs. And then 2 novel viruses have been found infecting ginger flower that we are going to characterize fully. So we're going to publish on those viral pathogens. We're going to continue with fungal pathogenicity. I still need to survey Maui and follow up on seminars with current knowledge. Now, I did produce a better management practice for preventing and controlling pathogens in red ginger. That is available. And if you want to email me at the end of my slides, I can send you this publication. It's fully finalized. We're just waiting to publish it. We're coming up to 20 minutes here. I kind of wanted to talk about Podocarpus blight a bit. I knew I put too many slides in. Just give me 5 minutes and we'll do question and answer after. So Podocarpus blight, it's an issue that's affecting Afrocarpus trees, multiple varieties. Symptoms tend to be shepherd's crooking, full plant decline. These are very indicative symptoms of Podocarpus blight, whereas symptoms for the other pest in Podocarpus, which is black twig borer, are isolated brown dying branches. Now, we did identify 3 major fungal pathogens, Pestalotia alternaria and Coletta trichum. It was found that mostly weak and wounded trees were the only trees that became infected with this mix of pathogens. You can see the pathogens here, just to prove that I had them on some plates at some point. I tried a bioassay in the lab. So it was a bioassay done on plates. I used 3 different fungicides, Caproxit, Heritage, and Manzate. All 3 are landscape and nursery available fungicides. We also did a control with plant exudates and no exudates. Just to run through quickly to show you some of the data on what we got. Tall bars are bad for efficacy. So anywhere where you don't see a tall bar, you saw efficacy. This is for Pestalotia. Basically what this shows is Caproxit and Manzate work the best. And plants that had plant exudate or plates that had plant exudate added saw more germination from the Pestalotia. Same is true for Alternaria, Caproxit, Manzate, really the only fungicides that worked. And again, Caproxit, Manzate worked well, Heritage also worked well in this case, whereas no control leads to a lot of germination. So for this one, in conclusion, weak and damaged Afrocarpus are more susceptible to blight. I've only seen it on Oahu. I have not seen it on Big Island yet because plants are often not grown in areas where they shouldn't be grown, essentially, which is very close to the ocean, in very dry climates, or in climates where you've got parking lots and you've got the plants weakened through drought and sun damage, heat damage. Try to maintain healthy trees, avoid pruning in extreme weather, and Caproxit and Manzate are recommended. Now, this is important for the nursery and the floriculture industry as well, because Afrocarpus is a plant that's commonly used as either a windbreak, as a nursery crop, or as a cut foliage plant. I want to publish on pathogenicity, so if there's any takers, I still have isolates available. We have to identify the pathogens down to the species. And I'd like to do an in-field efficacy study to see if that bioassay holds true. Now, I was able to control them on my own using Caproxit in a nursery setting, but this wasn't a repeated study. So here's my email. If anybody wants that better management practice before it hits the books or before it hits the web, I want to thank HDOA because they helped out, first of all, with funding, and then also Manco was essential in doing the Podocarpus blight identification and bioassay work. He let me work in his lab. I also want to thank my collaborators for the Ginger project. Thank you. I'll take questions.
We do have a question here for you.
That's good.
It says here, per ginger virus, have you tested any non-symptomatic plants for the virus, for viruses?
Yeah, so as you saw, okay, non-symptomatic plants. So we have tested some non-symptomatic plants, yes, but none had the virus. We're actually trying to find non-virus plants right now because we want to work on improving or developing stock of virus-free plants. So I'm trying to find non-virus plants currently. So also, if somebody thinks they have non-virus plants and they want to contribute to the ginger industry, contact me because I would love to test your plants to see if they have the virus. And if they don't, I would ask for a small sample so we could start doing tissue culture. I've got several roots. One of the other speakers on here, Dr. Fitch, and then we also she works in a tissue culture lab. She runs a tissue culture lab, and I want to work with them on doing multiplication of virus-free ginger. We also have a potential tissue culture lab here over on Big Island that can work on that, a private industry member.
Okay, so at this time, if someone wanted to get clean red ginger plants, that is not available?
So it's almost impossible to find nursery crops that are clean. What I've been recommending to growers is find a reliable and trustworthy friend. I think the best island to do it on would be Big Island, because we've seen the least amount of viral infected gingers on this island. And we haven't seen some of the more severe viruses like the nucleorhabdovirus. The only problem is these viruses are everywhere. So finding a clean plant is almost impossible, especially in nursery conditions, especially in landscape or field conditions, because of the uncontrolled and undocumented spread for multiple years before it was even identified as an issue. So the best thing to do is, if you're going to plant a big field, possibly just wait until there are clean gingers, or find somebody that has the least symptomatic plants out there. So the one thing you want to watch out for is that chlorotic banding and stunting. And any of those other symptoms I talked about are symptoms that you need to watch out for also. But unfortunately, no, there's no trustworthy source, to my knowledge right now, of clean ginger, which is unfortunate and nobody's fault, because this is just how it goes. But yeah.
So if a grower has his plants are his or her they're infected with the viruses, should they be rooting it out, disposing of it, or just get what they can out of it?
We go over that in the Better Management Practice Guide somewhat. At this point, I'm not recommending removing the plants, because most likely all the plants in that field, around that field, are also infected. It's a lot like some of these other, like for example, T virus. It's everywhere. And most likely the plants that you would get to replace it are going to also be virused. So trying to maintain your plants culturally as healthily as possible, proper fertilization, proper irrigation, proper soil management, reducing stress in the plants, and reducing insect pressures in the plants is going to help with still getting yield and production while still having the viral symptoms. Whereas there's really no solution when it comes to ripping them out, because most likely what you'll put back in are virused plants. So if you're going to rip them out and, for example, grow something else, maybe. But ginger is a product that we are seeing a decrease in availability due to a decrease in people producing it. So removing ginger is maybe not the economically best decision, because you will have the ability to market it and sell it.
So Russell, you're located on Hawaii Island right now. How often do you get to Oahu?
So I am supposed to go to Oahu once a month. Travel has made that difficult, but moving forward, it's once monthly. I'm going to Oahu, and that's dependent on funding. Yeah, but I am the agent that's also covering Oahu. So if I'm not there physically, you can contact me still, and we can arrange a visit when I have one of those scheduled trips over to Oahu. So the next one's going to be, I don't have it planned yet.
All right. Thank you very much, Russell. And I'm sure if more questions come in, we'll turn them over to you.
Well, if anybody has questions, contact Galanti at hawaii.edu, and we can talk a little bit more in depth. Thank you.
All right. Great. Thank you.